What defines who crosses the finish line first at the end of a race? Is it nutrition, effort, or perhaps something written in our DNA? Roughly 66% of the variance in athlete status in women could be attributed to genetic factors, suggesting that the race may begin at the molecular level. For maximum performance in each sport, many different physiological and biochemical systems must interact and function optimally. Each of these systems is influenced by the proteins that constitute them, encoded by our DNA. One such protein is MCT1. Let's explore how variations in genes like MCT1 can influence our chances of winning first place. Welcome to the genetic base of stuff and things. During exercise, both aerobic and anaerobic pathways fuel muscle contraction. Skeletal muscle consists of different muscle fiber types: white muscle fibers, which rely primarily on glycolytic metabolism, and red muscle fibers, specialized for oxidative pathways. How do these different fibers coordinate to meet the high energy demands of physical activity? The answer lies in lactate. Lactate's role has been largely misunderstood. In fact, many in the sports world still consider it an enemy. However, in 1985, George Brooks introduced the Lactate Shuttle, proving that lactate is not a simple metabolic waste product, but plays critical roles in the body. As we exercise, white muscle acts as the main producer of lactate, which must be cleared to maintain high rates of glycolysis and intracellular pH homeostasis. Excess lactate is shuttled within muscle tissue and in the bloodstream. Some is then taken up by red muscle fibers, where it is oxidized, supplying energy to the cells. In fact, lactate has proven to be a preferred respiratory fuel during exercise. But before lactate can be oxidized in red muscle fibers, it needs a way into the cell. How is this achieved? Enter MCT1, a member of the monocarboxylate transporter family, responsible for the co-transport of protons and monocarboxylates like lactate. MCT1 is found in several tissues. In a skeletal muscle, MCT1 is specifically located in red muscle. Now, here is where genetics come into play. A common variant of the MCT1 gene shows a single nucleotide change from adenine to thymine at position 1470. This results in an amino acid substitution in the MCT1 transporter at its C-terminal tail, from glutamate to aspartate. Because the C-terminal region is believed to influence substrate specificity, this variation could impact the transporter's affinity for lactate, limiting its rate of transport. In fact, this MCT1 variant was initially found in subjects with a 40% reduction in erythrocyte lactate transport. Researchers were then interested in investigating the influence of this MCT1 polymorphism on lactate dynamics in sports. A pilot study recruited 10 men aged between 20 and 26, with at least 1 year of regular resistance training experience. They were categorized as non-carriers if they showed 2 wild-type ALLs, or carriers if they show at least 1 MCT1 mutant TLL. All subjects underwent the same initial adaptation phase, and were tested on 3 different circuit weight training sessions on non-consecutive days. Each session involved a warm-up circuit and was at an increasing level of intensity, tailored to their individual strength level. The training protocol included 3 circuits of 8 standard exercises with short breaks in between. Capillary blood samples were obtained immediately after the completion of each circuit, and at minutes 3 and 5 after the end of each session. Blood lactate concentration was measured, and the change in lactate concentration during each session was calculated to measure lactate accumulation at each intensity. Carriers of the MCT1 mutant TLL show significantly higher blood lactate accumulation at the highest intensity being tested. The overall maximal blood lactate concentration reached was also higher in carriers. These results suggest that the MCT1 missense mutation may disrupt the function of the transporter, affecting lactate intake into red muscle cells for oxidation, and leading to higher levels of blood lactate accumulation. However, a difference in lactate accumulation was only observed at the highest intensity tested, suggesting that a blood lactate concentration threshold might be necessary to observe a difference between the MCT1 variants, above which the reduced capacity of the modified transporter starts to impact lactate accumulation. But this is only a pilot study. Further studies on bigger cohorts, including female subjects and diverse ethnicities, should be considered. In fact, the authors later investigated the effect of this polymorphism in women and observed no significant difference in the activity of the 2 MCT1 variants. This might be due to the lower blood lactate levels observed in women, which might be below the hypothesized threshold. How could all these findings relate to sports performance? Think of it as a relay race, where lactate is a baton, and MCT1 the runner handing it off to red muscle cells. Just as a smooth handoff can affect a relay team's victory, efficient transport by MCT1 could be critical for blood lactate clearance and energy metabolism. Any disruption, like a deficient variant of MCT1, may slow down the handoff, leading to blood lactate accumulation and reduce energy supply in red muscle. The importance of MCT1's role in exercise is reflected in various studies. Training has been shown to increase the concentration of MCT1 in human skeletal muscle. MCT1 content is also inversely correlated with fatigue indexes, suggesting that lactate uptake, its oxidation in red muscle, and the associated proton transport are significant for maintaining force production. However, genetic association studies looking at the link between the MCT1 genotype and specific athletic phenotypes report diverse results, making it hard to establish a direct association between this polymorphism and a specific athlete status. This highlights that someone's athletic ability cannot be entirely determined by their genetic makeup. Multiple genetic interactions, epigenetic contributions, and environmental factors also play key roles. Nevertheless, understanding our DNA can help us predict athletic performance and fine-tune our training plans to maximize results and prevent exercise-related injuries.